Bueno, buenas tardes a todas, a todos. Nuevamente les doy la bienvenida en nombre de la Comisión Directiva de la Sociedad Argentina de Inmunología en esta nueva serie de webinars que tienen un foco un tanto diferente a lo que estamos acostumbrados y que creo que nos es de gran utilidad a todos y todas los miembros de la sociedad, ya que muchos de nosotros, o la gran mayoría de nosotros, además de investigadores, somos docentes de grado y de posgrado en las universidades argentinas. Este webinar ha estado organizado por la Comisión de Docencia de nuestra sociedad, que realmente ha estado teniendo un rol sumamente activo, no solamente organizando las actividades del Día de la Inmunología, sino también les comento, porque realmente creo que es muy interesante, cuando ustedes entran a la página web, pueden ver en el banner de novedades que la comisión docente está incorporando permanentemente reflexiones y pensamientos sobre César Milstein en homenaje a su año, que ha sido decretado el año de César Milstein por un decreto nacional. En esta ocasión, también el comité docente se ha preocupado por organizar estas actividades, principalmente Rubén Motrich y Daniel González Maglio, quienes van a coordinar esta sesión, han invitado a expertos en educación en inmunología. En el día de hoy nos acompaña el doctor William Carr, quien va a ser posteriormente presentado por los coordinadores de la mesa. William, thank you so much for being with us today. For the Argentinian Society of Immunology, it's an honor, a pleasure to have you here sharing your experience in teaching immunology at different levels, and we are really eager to listen to your talk because I'm sure we can exchange a lot of experiences, and it will be really very exciting. So thank you again, and I will leave you now with Ruben and Daniel, who will chair the session.
Oh, thank you, Mariana. Do you listen to me?
Yes,
okay. Good afternoon to everybody. It is a real pleasure and an honor to introduce the speaker of this webinar, Dr. William Carr. Dr. Carr received his Bachelor of Science degree in zoology from Duke University at Durham, a doctorate of veterinary medicine from North Carolina State University, and a PhD in immunology from Stanford University, California. Currently, Dr. Carr is a full professor with tenure in the Department of Biology at Medgar Evers College, City University of New York at Brooklyn. His research interests include investigating innate immune response in HIV pathogenesis and associated co-infections. But in addition to that, he also investigates novel teaching and learning strategies among underrepresented minority undergraduate students. Since 2012, he has immunology lectures and laboratory courses, a general biology 1 course for biology majors, as well as a Principles of Virology course at Medgar Evers College. Remarkably, he currently serves on the Education Committee of the American Association of Immunologists. The title of his presentation is Using Pillars of Immunology Research Articles to Enhance Problem-Solving Skills Among Undergraduate Students. Dr. Carr, on behalf of the Argentinian Society of Immunology, thank you very much for accepting our invitation and sharing activities with us.
Oh, thank you. Thanks for inviting. Let me see if I can share the screen here. Oh, oops.
Yeah.
So can you see everything?
Perfectly. Yeah.
Good. So thanks for inviting me, and so I can share some of the work that I've been doing on kind of teaching innovations, particularly for teaching immunology to undergraduate students. And for today's seminar, I'm going to focus on the work I've been doing using the Pillars of Immunology research articles as teaching tools. So what I'll be doing today is talking about the Pillars of Immunology resources, as well as identifying, briefly talking about the impacts of this work on the teaching and learning outcomes for students, and how this can be implemented in teaching immunology through scaffolded exercises that can be performed both before and after implementing this teaching tool. And then talking about how you can assess this work, as well as give you some examples about how it can be implemented both in a small setting or a setting where you have individual students, also a setting where the students can do group work and work on this for several weeks at a time. And then also I'll be talking about some of the challenges in implementing this, as well as addressing questions that you may have. So, and to start off with, as educators, we want to teach our students critical learning skills, both so that they can apply the knowledge, as well as strategies for looking at information in a critical way. And it's been well documented that students learn more by doing than they do by listening to lectures or watching videos. And so this is just one application of students can learn by doing. And since interpreting data and designing experiments are really at the heart of a work in any STEM field, particularly in immunology, that these resources, the pillars of immunology, are a useful resource for students having the ability to practice analyzing data, and also to incorporate the fundamental principles of immunology while they do this in a supported environment of the classroom. So the pillars of immunology are a collection of over 200 research articles that are really part of the Journal of Immunology. And these articles have established paradigms in the field of immunology. And these are available through the website of the Journal of Immunology. And this collection of articles is searchable by topic. And what is one of the benefits of these articles is that the research articles come with an associated review article. And so the review article is important because it provides an overview of the field, as well as puts the research article into perspective, so that you get a sufficient background with the research article as well. So in using these articles as a tool to teach critical thinking skills, in surveying the students, I found that students that I used this approach were more confident in their ability to interpret data. And this was done through pre and post surveys, kind of surveys given before administering this, as well as after administering it. And then also, when I objectively look at their scores in how they performed on exam questions requiring analytical skills, I found that I saw dramatic improvements as well. Although the students didn't quite reach the mastery level in analyzing data, and I attributed this largely to many of them didn't have a background in many of the methods that were used in generating the data. And so since then I've supplemented the implementation with more exercises on the methods in particular, and how the methods are done to generate data. And I continue to collect additional data on the assessment and how to continually improve this method. So the idea or the focus of this particular teaching tool is to achieve the following learning outcomes, mainly to focus on data and data analysis, in that the students will be expected to design and evaluate the validity of experiments, which are based on hypotheses around immunological principles. And also to focus on interpreting primary data that were generated using immunological approaches, and in particular to identify the controls in these experiments, to identify positive controls and negative controls, and also, in addition, to work on kind of some more soft skills, which include working in a team to critique experimental data, and also to work on their oral communication skills, particularly communication about science. So to implement this particular approach, there are several different potential approaches that you can use. Most of these approaches depend on the time constraints and the mode of delivery. So if you only have a limited number or limited time available in a course to implement this, you can do this through a discussion board or individual homework assignments. And in this particular implementation, this is done by administering multiple choice questions focused around one data figure. And I'll give an example of this. And or if you have more time available in the course, I'll say 1 to 2 weeks for implementation, you can have the students work in small groups or work on virtual groups online, focused on a key data figure and for group presentation. So this can be done either in class or it can be done online. And then finally, if you have 2 or 3 weeks to implement this, you can give group assignments where the student groups present the entire research article in a format of a journal club. And depending on the class size, you can allocate maybe two class sessions for this devoted to the student presentations. So now what I want to discuss are some of the scaffolded assignments to both provide the foundation so the students can do the data analysis and know basically what the format of a research article is and why it's important to critically analyze data. And so one of the scaffolded assignments that I give early on, whether you're going to give a limited, say 1 week implementation or a longer 2 or 3 week implementation, is to give an assignment based on why data analysis is, well, why critical analysis of data is important. And so to do this, I give the students this article. This is a freely available article that is based on an actual study that was done evaluating the use of a parachute for people who did and did not use a parachute jumping out of an airplane. So it was a randomized controlled trial where they had one group that had the parachute and another group that didn't. And in this exercise, I give the students the article, and then I also give them 2 questions that they must answer. The first question is, would you jump from an airplane without a parachute based on the data in this paper, not on their opinions, but only on the data in the paper? And then the second question, on which data table or specific data are their decisions based on? So I give them the paper and the 2 questions and have them answer those questions. And I found that I get some very interesting answers to these questions, whether I do it as an in-class activity or do it as a discussion board online. And I find that many students realize in doing this that it's important to actually look at the data themselves. So the authors of this paper conclude that there's no difference in the groups and that it's okay to jump from an airplane without a parachute. But what the authors don't tell you is that you have to look at the data yourself and to see that in this particular trial, the velocity of the plane was 0 and that the people were jumping only about 0.6 meters or about 2 feet. So this were very kind of unique conditions. And so I highlight this to the students as they go through the paper and we discuss it in class or discuss it online, that it's important for them to critically analyze the data themselves. And this is how I kind of start off the course so that they first appreciate that they need to do their own critical analysis of data. And then to provide additional background and methods, I include some virtual labs for them to get experience on key laboratory approaches or techniques used in immunology. And one of these is a virtual lab on flow cytometry. So the website that I show here is a website that's freely available that I think is provided by the BD Biosciences. And this is a site where they have several different components that you can go through, and each component has a quiz at the end that tests your ability on understanding different components of flow cytometry, and it goes through various details of not only how flow cytometry works, but then also they talk about compensation, and they talk about some of the problems that you may have, and also the applications of flow cytometry as well. And so I find that this lab is a useful introduction to flow cytometry, and so I give this early on in the course for the students. A second thing that I give to the students is an assignment where they have to actually look at a research article that's been annotated. So this annotated research article allows them to appreciate the different components of a research article, and also it gives them further practice in understanding research methods, because this annotated article annotates the different components of the article, of a research article, and it gives some additional details about why experiments were done and what controls were included in those experiments, as well as it allows the students to highlight different sections of an article to see where the information in the article can be found. And so I found that this is a very useful activity. And I usually give it as a group assignment because there's some students who were able to catch on ideas quicker than others. So in this way, the students can work together and learn from each other to learn about the different components of a research article and how to read a research article. And then another component that I include as a scaffolded exercise is a virtual lab on ELISA. And I found that this virtual lab is sponsored by the HHMI, and it's a very useful lab because it allows, the focus of this lab is on experimental controls and also data interpretation. So in this particular virtual lab, the students do an experiment virtually and they actually get results. And then they have to interpret those results. And if they don't do the experiment correctly, they'll get some erroneous results, and then they have to explain why those results turned out the way they did. And so I find that this lab is very useful to students to get kind of virtual hands-on experience on how to do an experiment and how things can go wrong in an experiment. And also this particular exercise emphasizes why it's important to have positive and negative controls. So they get additional experience in doing that. And then for a typical 15-week course, about in the middle of the course is when I would implement this particular innovation or this intervention. And in doing an analysis of the Pillars of Immunology articles, I use 4 key areas to emphasize. One is including the background or context of the work, which is include the method or approach used. A second area is what are the negative controls that were used, and most importantly, to explain why those controls were included. Likewise, another area that I have is on positive controls and explaining why those were included, and then trying to extend the work by proposing additional controls or alternative approaches that could be used. And this can apply to a whole paper or it can apply to just one figure. And so that's why I found that this particular approach is very useful, because it is very versatile in applying to just one figure or to applying to, the students can go through a whole publication applying the same 4 areas of concentration. And in addition, if I'm using this exercise for a, so this is the exercise using it for a small or limited application. So if you only have, say, a week to implement it, you want to focus on one article. So here I give one example of a research article on basically how they discovered the role of FOXP3 in the development of regulatory T cells. And so in giving the article, I first give them the review article, which is the one done by Jeff Bluestone. And then rather than giving them the entire article, I'll give them, say, if it's a limited time available, I'll just give them a key figure from the paper. And so in most cases, the key figure from the paper is going to be, there's usually about the second or third figure in the paper. And this is the key figure because this is the figure that gives the whole kind of take-home message from the paper. And so I'll pick out that figure with this legend, and I'll give that to the students, and I'll also give them the review article, and then I'll give them some questions. And so this I'll implement after I've covered the material in class, whether it's a lecture or provided them with videos covering the material. So in this case, I'll administer this after.
Se cayó la...
Sí, esperemos un minutito, seguro que se
Un
vuelve a
minuto,
conectar.
se puede conectar. Mientras tanto, podemos decirle a toda la gente que cualquier duda, pregunta o comentario que vayan teniendo lo vayan poniendo en el chat, así nosotros después podemos hacer bien interactiva la discusión con él.
Sí, creo que hasta que él se vuelva a conectar podemos ir charlando nosotros sobre cosas que le podríamos ir preguntando. A mí me parece interesante comentar un poco a la audiencia que está toda conectada en YouTube algunos de los aspectos que estuvimos discutiendo. A mí me gustaría saber, por ejemplo, el tamaño de las clases que tienen ellos, si son semejantes a las clases que tenemos en Argentina, que son bastante masivas nuestros grupos de alumnos. También estuvimos charlando un poco sobre distintos aspectos de cómo
Sobre el
ellos
background
el
sobre los
background.
diferentes backgrounds que por ahí tienen los alumnos.
Ujú, exactamente. Bueno, todo ese tipo de cosas que quizás pareciera que estamos en dos mundos totalmente distintos, por ahí no son tan diferentes. Estaría bueno poder charlarlo, comentarlo. Así que invitamos a todos en el canal YouTube
Sorry
que
about
vayan colgando sus
Yep.
pensamientos. Ahí está
Yeah,
William.
William.
Sorry that I.
Yes, we are watching your presentation, William. It's okay.
Okay, sorry that I just my power cord came out. So after doing that, after implementing or actually giving a lecture or supplementary videos on kind of T cell maturation and T cell differentiation, and specifically including information about how CD25 can be used to identify regulatory T cells. And also after giving the virtual labs on the ELISA and flow cytometry, I implement this as a homework or in-class small group activity. And I also mentioned this before, I provide the review article and then I also provide the figure 3, which is a key figure of this paper. And so this is the figure 3 from the paper. And in figure 3, it shows a key experiment that they did where they basically took cells that were not regulatory T cells and they co-cultured them with cells that they had transfect with or without the FOXP3 transgene and showed that this transfection of the transgene allowed the cells to have the function of depressing the secretion of IL-2 activation by the cells. And so it shows the various controls here and it shows the results. And then I, again, focus on those 4 areas, which are the methods, negative controls, positive controls, and then advancing the study that was done. And for each of those points, I emphasize that the students need to explain why their answer was correct. And so in terms of administering it as a discussion board, I would give the figure, give the supplementary documents such as the review article, and provide those scaffolded assignments, and then give them the question of, in this experiment, what was the method that was used? And so the students will then answer that and then give an explanation of why their choice is correct. And so in this case, it would be the ELISA of the culture supernatants, and it's because they're measuring or quantifying the amount of IL-2. And so that is the appropriate answer for this particular question. And so this would address the kind of question about background. I would also, as a second question, give them a question about the negative control. And then negative control here, prior to this, I would also explain what a negative control is, and then ask them to answer that question. And in this case, the answer would be that the CD25 negative cells alone and the cells that were transfected or transduced with an empty vector, which is a +1 GR1, those would be the negative controls, and explaining that those are the negative controls and really emphasizing that they need to justify their answer to explain why those are the negative controls. And then as a review, we could go over it and talk about why this is a negative control and how it shows why it's important for this particular experiment. And then I would also ask them about positive controls. And then I would identify the same parameters and say which of these is a positive control. And the answer would be the CD25 positive cells. So these are regulatory T cells identified by expression of CD25. And then they can also see that administration or co-culture with these cells has a dramatic effect of reducing the production of IL-2 by the other cells. And so they would also justify why this is a positive control. And then finally, I would give them a question, multiple choice question. So this is multiple choice because it's easiest to administer in the discussion board, and also give them opportunity to explain why their selection is correct. And in this case, I would present different possibilities of how they could make this experiment better, or give them more confidence in understanding that the experiment is working the way that you would predict. And in this case, the answer would be by taking the FOXP3 transgene and putting it into CD8 T cells, because they know that CD8 T cells are not regulatory T cells. Regulatory T cells are only CD4 T cells. And so by doing this and basically finding that they wouldn't have an effect, it would give them additional confidence in the data. So in terms of what experiment they could do to kind of extend the data, this allows the students to be more creative. If you're giving this as a group activity where the students kind of write free form, or where the students can give a presentation, it allows them to be more creative, and also allows them to bring in more current techniques. So many of the studies that were in the repository of the Pillars of Immunology are older studies. And so since then, many newer techniques have been developed that can address the same questions. And so it gives a chance for the students to kind of think outside the box and be more creative. And so in giving this to students as a group activity for a group presentation, what I found helpful to do is to kind of pre-select the articles, because many of the articles may have more complicated methods, or some of the articles may cover topics that have not yet been discussed in your course. So I pre-select the articles and to limit them to about 7 or 8 potential articles. And then I allow the students to choose which article their group wants to work on. And I find that this selection process offers the students some agency in kind of being more motivated in presenting the article that they chose to present, because they chose to present that particular article. And then I give it them with sufficient time so they have at least 2 weeks to prepare for their presentations. And again, I provide them with a companion review article with each review article. And then also I allow the students to schedule a 30-minute question and answer session with me, kind of outside of class time, so that any questions that may have about the article can be addressed before they give their presentation. And I found that many times the questions that the students have are related to the methods or approaches that are used. And so it gives me time with them to clarify that material. And this gives you kind of an example of a range of articles that have been used previously in the course to kind of convey these ideas. And the article that's highlighted in yellow at the top is the article that I did discussion of as an example for the discussion board that I went through with the 4 different questions. And this was again on the regulatory T cells. And as you can see from the list of articles, it covers a range of topics from innate immunity with the toll-like receptors through the development of Th1 and Th2 paradigms through a range of different topics in immunology. And each of these has a key figure that you can pull out and use for teaching specific areas or for limited use, as well as using the whole article for the teaching purposes. And so when I give the articles to the students or have them pick an article, I also give them specific guidelines and instructions of how they should give their presentation. So the instructions that I give them include the following: that they should discuss the work and put it into perspective of what was done before, or how this was transformative to the field, and also to mention what methods were used in generating the data that they reviewed. And specifically, when they discuss the data, I asked them to talk about the controls that were used, to explain why the controls were included, were they positive controls or negative controls, and also just to tell what the figure shows, as you would in a journal club presentation. And also I emphasize that each of the group members should present at least 1 figure. So that's why I have the group members limited to 4 or 5 students per group, so that each person can present at least 1 figure, and also have them propose how the overall work can be improved or strengthened. And this can also include what suggestions they think should make, and to emphasize to make suggestions that are more transformative than kind of minor suggestions. And then also as part of their grade of the assignment or score for the assignment, they also are encouraged to elicit questions from their peers in the audience. And so this brings in some interaction with their peers so that they have a interaction in learning the material as well. And so again, each group member should present at least one figure. And I tell them they can use PowerPoint slides, or they can use a whiteboard or another presentation platform to give their presentation. And for a typical, say, hour and 15-minute class, I tell them that the presentations are limited to 20 minutes with 5 minutes for questions. And again, to emphasize that they should elicit questions from their peers because that's part of their grading rubric. And then, as I mentioned before, that they can schedule a 30-minute session with me prior to their presentation to address any outstanding questions that they may have. And then prior to having those group presentations, I also give a presentation myself to show how it can be done. And the article that I present is one on flow cytometry. This is one of Herzenberg's early works on how the flow cytometry works. And so I present this, I give them both the review article and I give them the actual article. And then I go through the actual article as I would have them present the article. In other words, I emphasize the controls as I go through the presentation and explain why the different controls are included. And also I give some suggestions as how the work could be improved. And this is more to model the behavior so the students then know what they should aspire to and how to go about presenting the article as you would in the journal club. And as a final note, in terms of getting the students to be accountable to each other, I also have them, after the presentation is done, I have them rate the participation of each of their fellow group members. And then that score that they rate their fellow group members is used to add as bonus points to their group score. So each individual will receive the same group score from their group, but then they will receive additional points based on their participation average score that they receive from their fellow group members. And to finish off, I'll give them an assignment of a one-page self-reflection. So this assignment is more getting them to reflect on their activity in the group and their participation in the group and their role in the group. And then this self-reflection assignment, I also give them a rubric to work from so that then they can then go through and carefully think about their role in the group and also what areas of group work they may want to work on in the future. And so this is an example of a typical teamwork rubric that I use, where it gives you some different areas and how the different areas rate in terms of contribution, problem solving, their attitude, keeping people focused on the task, and working with others. And it gives them kind of guidelines as to how they would rate themselves, whether it's a 1, 2, 3, or 4. And I have to ask them to go through this and include this in their self-reflection, kind of one-page end of the assignment activity. And this gives them a chance to kind of reflect on their role in the group and think about some of the non-hard skills, more soft skills in working with others. And so in terms of assessing the learning from this activity, again, for the activity, I select the key figure and legend from each paper that's presented and then include them on a midterm or final exam. So I pick one with the key figures from the papers that are presented, have them as a part of a question of the final exam, and I have them choose whichever figure they want to ask questions about. And then I give them the same 4 questions about explaining what method was used to generate the data, identifying the negative control and explaining why it was included, identifying positive control, why it's included, and then proposing additional controls or alternate approaches that can be used to strengthen the conclusions of that particular figure. And so then that can be one way to kind of objectively assess the learning of the activity. And then in terms of what challenges or issues I found that the students had, so one of the issues I found that I mentioned early on was the students were not familiar with many of the methods that were involving molecular biology or cell biology techniques. And so what I've done since then is to include supplementary videos on these techniques to better explain how they're performed and to give them more practice in this particular activity. And then providing them additional references, such as the appendix of many of the common immunology textbooks that discuss experimental methods, so the students can get a better foundation in methods that are used in many of the papers. I also found that some students didn't understand how to read graphs. And so this I've worked on by starting off early in the course, including opportunities to interpret data. And so in a typical lecture or discussion, or even in an active learning group activity as part of a class, I'll give them some data to interpret or graphs to interpret. So then they get more and more practice on how to interpret graphs and how to interpret different types of data, whether it's flow cytometry data, whether it's line graphs or bar graphs, to give them practice in this area before they actually have to present paper. And with that, before I finish off, I just wanted to highlight that if you're going to be attending the virtual immunology coming up next week, that I'm going to be running a breakout session on using another tool for teaching. And this is a freely available online platform called H5P. And this is, I've found exciting results using this tool for teaching difficult or challenging immunology topics like somatic recombination or VDJ recombination. And so I'll be talking about that in the breakout session, as well as I have a poster covering the same topic. And you can find the poster by just searching for my name in the poster materials. So with that, I want to open up for your questions, and thank you again for inviting me to speak.
Thank you, Dr. Really nice to listen to your very interesting and enlightening presentation to your talk. So the presentation of Dr. Carr is open to questions. We have some already. And the first one is, if in the scaffold building, does the order of the subjects matter?
So I find that the way that I have done it is first for starting, I think starting from the bigger picture of why it's important is to present first, and then after presenting why it's important is to presenting different strategies and give the students practice on how to do it. Because I found that many of the students don't really appreciate why it's important to be critical of analyzing data, because I found that students often kind of take what's written as being true, but not necessarily questioning that. So first, kind of breaking that myth is important before they can start to kind of appreciate why it's important to be critical.
Mm-hmm. Daniel?
Yes, I have a personal question. Do your students have experience in previous subjects working with these kind of research articles in different subjects?
So I find that many of the students don't. It's their first time really reading a scientific article. And many of them, when they start the course, are intimidated by articles. But I think going through exercises of kind of a figure by figure, the students by the end feel much more comfortable in approaching articles and realizing that they can do this and they can get through it.
Another question is, how big is your classroom? How many students do you have in your classroom?
So typically my class is about 20 students or less, but in the pandemic, class has gone virtual, so I have up to 40 students. And one difference I found that in working with a larger class is I found it even more important to have the students work in groups, both for the benefit of the students as well as for the benefit of my grading load.
Mm-hmm. And do you have any experience with these tools with students who don't have English, or who do you know that their mother tongue is not English?
Yeah, so many, well, some of the students I have, I know that their mother tongue is not English, some are Spanish, and some are French. And I find that what is helpful in that case is, again, having the students work in groups, because many times when the students have questions, they'll more likely to ask another student than they are to me. And also I try to often to restate as much as I can different ways of wording a question, and I try not to use abbreviations when I can, or if I do use abbreviations, to write them out so that the students don't have to struggle with that too much.
Yes, because a challenge that we usually face here is that most undergraduate students they don't like reading papers in English. So sometimes it's not too easy to us to achieve this kind of activities.
Yeah, I think it's kind of starting off at a simple level of approaching it kind of as analyzing figure, and even you can reduce the figure to removing as much text as possible and just having the data, and having the students go through the exercise of interpreting the data and identifying controls in the data, because that is not limited so much to language.
Yeah.
Okay, Dr. Carr, do you make an evaluation of the students' background to organize the topics to work on each semester?
So what I find, actually I find that I almost have done that inadvertently, because I start off the semester by having the students kind of each introduce themselves through a discussion board. And in that discussion board introduction, I also have a question of, why are you taking this course, or how do you see this course fitting into your career plans? And so I get various answers from that. And so from those answers, it kind of gives me kind of a leading as to how to direct the course. Sometimes I'll have a course that I'll direct it more towards research, or have a course directed more towards medical applications. And I find that those questions early on in the semester kind of give me a way as to kind of structure the course. Because if I have a course where many of the students are thinking about going to medical school, then I tend to include more examples and also include more case studies in the approach. And even if I include research techniques, I show how they're relevant.
Dr. Carr, when you start these activities, do the students have a previous general background on immunology or not? Or
No.
if they don't, how do you manage with the differences in the background of the students?
So I find, for this course, the prerequisites are a general biology course. And so when I find that there are students who are weak in their general biology, then I kind of identify them early on in the course. Previously, what I've done is I've given them kind of pre-survey questions. And these pre-survey questions include just some basic principles of biology. And if I find that if they aren't getting those principles in the pre-survey, then I kind of pay attention to those students and see how they do early on. If they tend to struggle, then I meet with them and I tell them how they should supplement their studies with kind of refreshing some of the basic biology principles.
Dr. Carr, you've mentioned that you ask the students to rate the other members of the group. Is this rating anonymous?
So it is anonymous, but one thing that I found is that the students will, you get a variety of students. Some will collaborate with the other students so they all get the same rating, and others will rate them just from themselves and keep it to themselves. So it all depends.
Regarding that questions, the marks of the students are similar to those of the teachers?
So it depends. I found that most of the, so when they're rating it, they're more looking at their participation in the group. And you can kind of see it in the group presentations where the students, you can tell from how they present whether they prepared or not. And then you can see that in how they rate their fellow group members, where the ones that didn't score so well were also the ones that didn't seem to be very well prepared in the presentation.
So we have lots of congratulation messages. All the people are very thankful for your presentation and asking many questions. And one of them is how often do you have classes? How many classes per week do you have?
So
to the same classroom.
Yeah. So when we meet in person, the lecture class meets twice a week and the lab class meets once a week. For the virtual class, everything has been asynchronous. So for that class, I give one recitation, which is 30 minutes, and then I have about half an hour or an hour for group activities in that recitation session. And that's once a week.
How many time do you devote to preparing classes during the week, considering that you are also a researcher?
Yeah, so fortunately, what I did was for the asynchronous class, it took a lot of work early on before the semester started to prepare the course and basically prepare all the assignments. But then once that's done, it's just a matter of kind of monitoring it and to kind of do the grading. So it's much more manageable where it gives you time to do other things.
Yes, after listening and watching your presentation, it's like we don't live in so different worlds.
Yeah.
Any further questions? We encourage people to go ahead and ask.
Everybody is thanking for this opportunity of sharing Dr. Carr's experience and also the initiative of organizing this type of webinar. So I think the audience is very happy with this talk, really.
Oh, good.
Mm-hmm.
So if we don't have any further questions, I would like to personally thank Dr. Carr for being so nice and accepting our invitation at first. So thank you so much. This was a terrific webinar and a very good experience in my very own opinion.
Oh, thank you. Thank you very much for inviting me, and it's been a pleasure getting the questions.
I hope we can see you one day in person in one of our universities here, which have a very large amount of students. So sometimes it's hard to implement some of these approaches because our groups of students are really large, which has advantages and disadvantages, really, but okay. Mm-hmm. Hope we can meet you in person someday.
Yes, I guess I would like that.
And you are mostly invited to come to visit Argentina.
I like that a lot.
Okay.
Hope to see you soon here.
Okay, thanks very much.
Okay.
Have a good evening.
Thank you.
Thank you.
Hello.